Please take your seats. Welcome to the first plenary session. The ECTH is not only about thrombosis, but it's also about a little bit more than thrombosis. And for that reason, we have invited a guest for our plenary lecture. This touches upon our field, but it isn't thrombosis. It is so much more. It is meta-thrombosis, perhaps. We have invited a professor of neurology and translational neuroscience, Professor Malcolm Macleod from Edinburgh, and he has, next to his research in neuroscience, an interest in quality of science, or perhaps I should say the lack of quality in science. This is perhaps best seen in his inaugural lecture, the title, which was How Bad Research is Killing Science, or the fact that he was a lead author in the Lancet series on research waste. Without further much ado, Professor Malcolm Macleod.
Well, thanks, Bob, and thank you very much for the invitation to come along here and speak at this very interesting meeting. I do have a slight interest in thrombosis in that sometimes I use a drug called TPA, which I might tell you a little bit about later. But I want to talk to you about what I've learned in neurology and the neurosciences about how we can improve the value and increase the quality of the research that we do. Before I start, I should give you my acknowledgements of my collaborators, because I always forget at the end. Tracy Emin is a British artist who had a piece of an installation, an unmade bed, which you might have seen, which she called All the People I've Ever Slept With. And these are all the people I've ever written with, apart from this lady up here, Lindsay Thompson, who I have written with, but I'm also married to, so she falls into both categories. These are my disclosures. I'm a member of the UK MHRA Commission for Human Medicines, which approves new drugs for use in the UK, and the European Medicines Agency Neurology Special Advisory Group. I'm a member of the UK group that regulates the use of animals in research, and sit on a couple of committees, one from the Cure HDI Institute, their independent statistical committee, and on behalf of the Wellcome Trust, I sit on a pharma research steering group. So that's my disclosures and where I'm coming from. So I want to tell you a little bit about how I think research goes wrong and how we can put it right. This is a road sign from Wales, which some of you will know is one of the countries which make up the United Kingdom. We have some road signs like this in Scotland as well, where we have a native language. So in the top it says what the road sign says in English, and then underneath it says the same thing in Welsh. Except what this road sign says is, I am not in the office at the moment, please send any work to be translated. So the person who was using this tool didn't understand what was in the black box. They sent their data away to the black box, and they got some data out of the black box, and they just assumed that it was correct to the extent that they put it on a road sign and were much mocked ever since. This is an example closer to home. There's a technique that we use in neuroscience called functional MRI scanning. And that rests on the basis that if you use part of your brain, it's more metabolically active, and that causes a reflex vasodilatation that one can detect in an MRI scan. But as you might imagine, these scans are very temperamental. They require a lot of setting up and a lot of trimming. And so this group in Boston tried to improve the quality of their work by setting up their machine every Monday morning. And they had a competition in the group to see who could find the most unusual piece of organic material to put in the bore of the scanner every Monday morning. And one morning it was Craig's turn, and as he was on his way to work, he suddenly realized that he'd forgotten to get something for the scanner. Now, fortunately, his route to work took him past the Boston fish market, and he realized that no one had had a fish in the scanner before. So he stopped at the fish market and bought a fish, and they set the scanner up and they twiddled the knobs, and it was all set up to go, but then their healthy human volunteer didn't show. And his genius was to do the same test on the salmon. So one mature Atlantic salmon participated in the fMRI study. It was 18 inches long, weighed 3.8 pounds. They didn't know whether it was male or female, but they thought it wasn't important because it was dead. And then the task administered to the salmon completed an open-ended mentalizing task. So the salmon was shown a series of photographs of human subjects with a specified emotional valence. The humans were happy or sad or perplexed as to what was happening. And the salmon was asked to determine which emotion the human in the picture was experiencing at the time. Several active voxels in the rat's and the salmon's brain cavity lit up at a cluster level of significance of less than 1 in 1,000. And as they conclude, either we've stumbled onto a rather amazing discovery in terms of post-mortem ichthyological cognition, that's to say that dead salmon can think, or there's something wrong with the statistical approaches which they used, for which they were rightly awarded the Ig Nobel Prize in Neuroscience in 2012. Coming closer to home, I did my PhD largely in cell biology, and one of the techniques that was in common use at that time was using gene array studies. Where you were trying to understand a network, a sequence of events that would lead from some stimulus, in this case to neuronal survival or neuronal death. This cartoon clearly drawn by someone who's very interested in one of the cyclin-dependent kinases and not in much else. And one of my colleagues in Edinburgh, Ruth Dayton, was interested in cell death in a neuroblastoma cell line, the SHSY5Y cell line, in response to staurosporin, the PKC inhibitor. So she exposed some of the cells to staurosporin and extracted the mRNA, and had some cells exposed to vehicle and extracted the mRNA, and found the expression of these two populations of mRNA species and found the ones that were significantly different following staurosporin challenge. And then she sent them away to something called ingenuity pathway analysis, which tells you how these are linked together, what pathway is turned on by this cell death. And when she got the answers back, she was confused because it didn't really fit with any of her understanding of which biologies might be in play. And this is where she had a moment of genius, because what she did was she went to the gene banks and she selected 13 genes at random and sent them away to IPA, and then did the same thing with another 13 random genes, and again and again and again, 1,000 times with 1,000 randomly selected sets of 13 genes, and she got the IPA scores for them. And these are her data. So the IPA score is the negative logarithm of the odds of those genes being associated together by chance alone. Not one of her 1,000 scored at less than 10 to the minus 4. The median score was 1 in 1 million, and the highest score was 10 to the minus 36. So what this tells you is that genes are associated together just because they happen to be in the same organism and in the same cells, assuming some similar functions, but it doesn't tell you anything about how they're functionally linked together. And those of you who are my age will have seen many, many grant applications which have then been trying to follow up on these findings from gene array studies, which it turns out have got no meaning whatsoever. So closer to my own interests now, this is experiments in rat focal cerebral ischemia. What one does is occlude the middle cerebral artery in a rat, give it the drug of choice in which you're interested. Is that a bit better? My apologies. Give it the drug of choice in which you're interested and see whether you can reduce the volume of brain injury which is caused. And this is a poster I saw at the Society for Neuroscience in 1997, where they've given half dose or full dose, low dose glutamate, and they see a significant improvement in infarct volume. But when I tell you that half dose, low dose glutamate is 10 to the minus 60 molar, 10 to the minus 120 molar, and full dose, low dose glutamate is only 10 to the minus 60 molar, those of you who remember Avogadro's constant, 6 times 10 to the minus 23, will appreciate that there isn't actually any glutamate in either this column or in this column. So either there's something wrong with this experimental approach, or homeopathy works. And I don't think homeopathy works. And if I were a homeopath, of course, I would much prefer to see 10 to the minus 120 molar being a much more powerful treatment, because that's the percepts and the tenets of their belief system. So largely as a result of trying to illustrate some problems with the way research was done with in vivo research involving animals, I was invited to join the team for this series of articles in The Lancet published in 2014, led really by Ian Chalmers and Paul Glasziou, which I think, although they're quite a meaty read, they're a very important read for people embarking on a career in biomedical research. And we identified 5 areas, 5 stages of research at which waste could occur, where there were opportunities to increase value. Whether the questions that we asked were relevant to users of research, whether we had appropriate research design, conduct and analysis, efficient research regulation and delivery, accessible and full research reports, and unbiased and usable research reports. And I want to take you through some examples under each of these headings. So in terms of setting research priorities, you can think about research occurring on two gradients, on two spectra, where you've got research which might be relevant to advancing our knowledge, and research which is relevant to immediate application. So research in what we call the dull quadrant might not advance our knowledge very much, but it addresses important practical questions. Research in the Curie quadrant is pure basic research without consideration of relevance to practical issues at the present time, but in the hope that in time it might tell us something useful. And in the Pasteur quadrant here, use-inspired basic research to address important practical, in our case, clinical questions. And of course, where you don't want to be is in the waste quadrant, where you're doing research which doesn't really either advance knowledge or have any application. And there's quite a lot of research, unfortunately, in that quadrant. Let us illustrate that with an example from Ian Idis on the inefficiency of basic science research, where he looked at more than 25,000 reports in 6 basic science journals in the late 1970s, early 1980s, and found that 100 of them claimed that their discoveries had got clear clinical potential ready for immediate application. 20 years later, only 5 of those 100 had resulted in licensed clinical interventions, and only 1 of those was in widespread clinical use. So although the authors thought that they were doing great work here, actually very little of it translated into something that I, as a practicing clinician, could actually use. And the other thing which Ian Chalmers and others have identified is this problem about whose research priorities we address. So if you look at research which might involve research into education and training, service delivery, psychological interventions and the like in green, radiotherapy, surgery, perioperative interventions in red, and drugs, vaccines and biologicals in blue, and if you ask patients and their carers what kind of research they think is important, they think that education and training, psychological interventions, service delivery, and the like should account for more than 50% of our research. But if you look at registered non-commercial trials, or worse still at registered commercial trials, then the research that we do doesn't reflect the priorities of the patients that we do it to serve. Let's move on to the way that we actually do the research. Some years ago, I was interested in identifying a drug that was safe, that might work in experimental stroke, so that we could test it in the back of an ambulance. We know that time to treatment is very important with ischemic brain injury, and so the faster you can initiate treatment, the better. And we identified over 1,000 compounds that had been tested in the laboratory in a model of stroke. Of those, 600 had been tested in our favorite rat middle cerebral artery occlusion model, of which remarkably almost 400 improved outcome in that model. Of these, 97 only had been tested in clinical trial, and of those tested in clinical trial, only 1, clot-busting treatment with tissue plasminogen activator, improved outcome in those clinical trials. And going back to the basis for those early trials of TPA and stroke, they were mounted not because of these animal data, although the animal data are supportive, but because it worked in human myocardial infarction, the sort of cognate condition in a different organ system. And interestingly, we've got 3 treatments which work in human stroke: aspirin, decompressive hemicraniectomy, and stroke unit care, which weren't developed at all through animals. So in my field as a stroke researcher, our animal researchers, myself included, have failed dismally in bringing new treatments to our human population. We're not the only ones. These are 2 sets of data, one from Bayer in Germany and one from Amgen, where they looked at, so Bayer as the drug company, their model of drug discovery is to try and identify things in the literature which might be druggable, which they could exploit with a target molecule. And then the first stage of that is to try and replicate the literature findings in-house. And what Bayer found over 4 years and 4 different disease systems was that they could only replicate in full or in part 1/3 of the findings. And Amgen, looking at drugs tested in models which might be relevant to the development of cancer therapeutics, found that only 1 in 9 of the experiments could they replicate in-house. And this has led on to the replication crisis that people talk about in vivo research, which I'll talk about a bit more later on. So when we looked at those 1,000 drugs for stroke, of which none really seemed to work in human stroke, that sparked for me an interest in what the problem was between the laboratory and the clinic, and how we might try and understand that problem better. And at about that time, a company called AstraZeneca had just finished testing a drug called NXY059, a free radical scavenger, to see whether it might improve outcome in stroke. They'd tested it in 5,500 patients and it didn't work. They were so convinced, however, that the trial was going to be positive that they had marketed the drug, including its name, Cerevive, it helps your cerebrum survive, and even sent reps round to ask me whether I preferred the blue tablet colour or the purple tablet colour. And then the clinical trial came out and it was negative. And they lost 17% of their share value in 2 days, and it took them 7 years to recover, and it cost them over $10 billion US. So we went back to look at the animal data on which those trials were based. And here you have 11 publications, 30 experiments, 408 animals. Not very many animals, actually, it turns out. Certainly not enough. In these systematic review meta-analyses, when I show them, the grey bar in the background is the overall estimate of how good the drug is, what the effect is, and its 95% confidence limits. But when we split the studies into those that randomized, those that blinded the conduct of the experiment, those that blinded the assessment of outcome, those high-quality studies show substantially and significantly lower estimates of efficacy in those animal models. Not one of those 11 publications did all three of these things, and the two that were used by Astra to convince clinicians like me to put patients into the trial did none of those things. So with the benefit of hindsight, it's not surprising that the trial was neutral. So this was 2008. Let me take you back 45 years before then to Robert Rosenthal. And this is a critical publication which I think we've completely lost sight of, much to our chagrin. He was teaching graduate psychology. And in the last term of the one-year course, the students had to do an experiment. And in the last week, they did an experiment that he set up for them. And the experiment was testing cognitive performance in two cohorts of animals. One were maize-bright animals, which had been bred over many generations at Berkeley, which at that time was a great center for cognition research, had been bred over many generations to perform well in maze tasks like this, although they were naive to the task in question. And the other cohort were their slightly dumber bred and inbred cousins who performed badly in maze tasks such as this. And the task was pretty straightforward. The rat was put in the stem of a tea maze. One arm was always lit, one was dark. The dark arm was always reinforced with food, and which was dark and which was bright was flipped according to a predetermined random schedule. So you put the animal in. If the animal learns, rather like the junior postdoc, it turns to the dark side and gets a reward. And this is what happened. The maze-bright animals started off performing better in the task than the maze-dull animals, and over time there was an improvement in both cohorts of animals, but the improvement was much more in the maze-bright animals. And when he asked the students what they thought of their animals, these maze-bright animals were sort of bright-eyed and they'd got wet noses, the sort of animal you'd like to take home to meet your mother. And the maze-dull animals were surly and insolent, and in fact, 3 of the students were bitten by the maze-dull animals. And of course, the takeaway is that he'd done an experiment which you wouldn't be allowed to do just now, because the experiment didn't involve rat subjects, it involved human subjects. The rats were drawn at random from the same cages in the same animal house on the Monday morning that all of this started, and the only difference in the animals, including their biting behavior, was in the minds of the students doing the experiments and how their expectations transferred to the animal behaviors. And we've kind of forgotten that. So when we came out and said, oh, those NXY data for stroke were a lot of rubbish and it's no wonder the trial failed, everyone said, well, that's because you stroke doctors are stupid. You don't understand how to do proper research. And if you looked somewhere else in the neurosciences, you wouldn't find such a problem. So we looked somewhere else in the neurosciences. Here's the stroke data I told you about. Here's data for multiple sclerosis, blinded studies here giving us a lower estimate of efficacy. Alzheimer's disease, outcome measured in the Morris Water maze, blinded studies giving a substantially lower estimate of efficacy. This bioassay, as I was taught, of Parkinson's disease, the unilateral 6-hydroxydopamine lesioning, and then testing dopamine agonists, well, you only see the effect if you know the group to which the animals belong. So then we were told that, of course, neuroscientists are stupid. And if we were to ask proper scientists who'd got proper diseases, perhaps like thrombosis or hemostasis, or perhaps like cancer or HIV, where they had effective treatments, then we wouldn't see this problem. So we thought, where can we go? Where can we go to get research that everyone knows is the highest quality? Well, fortunately, the United Kingdom has a thing called the Research Assessment Exercise. And in 2008, it reviewed all the research submitted by all UK institutions. And when it considered the top 5 institutions for in vivo research involving animals, they concluded that we'd made an outstanding contribution to the internationally excellent position of the UK. Yeah, great, just as well now that we're on our own. And then they were impressed by the strengths within the basic neurosciences. And this didn't fit with what we thought was going on. So what we did was we went to those top 5 institutions and we got from PubMed every in vivo piece of research they'd published in the subsequent 2 years, 2009, 2010. And then we went to that piece of research and we found over 1,000. And we said, well, was it randomized? Was it blinded? Did it give you a sample size calculation? Did it tell you how and whether animals were excluded for analysis? These are the institutions individually for randomization and blinding and the like. They're color-coded to protect my future career. But you can see that the rate of randomization is about 15%, of blinding about 17%. Power calculations are woeful. There are significant differences between institutions. So if I was a national research funding agency, I'd want to use something like this rather than the impact factor of the journal in which the work was carried. More of that later. But if I tell you that during the same period, in a smaller sample from PubMed, a random sample from PubMed, the rate of randomization was 50%, our leading UK institutions are doing significantly less well than the average of PubMed. So there's a problem all over. And you can't go to a leading journal and say, well, because it's in Nature, or because it's in Cell, or because it's in Science, it's going to be fine. In our systematic reviews at the time that we did this work, published last year, we'd got risk of bias measures from about 2,600 publications for which we could identify the impact factor of the journal in which that work was published for the year in which that work was published. So what we did was we lined up those 2,600 publications by impact factor and split them into deciles, into tenths. And so here you have the reporting of a conflict of interest statement in the lowest 10% by impact factor, the highest 10% by impact factor. So that's quite good. That's probably because in vivo research, which gets into high impact factor journals, usually goes into a journal that knows about conflict of interest statements and so requires it. What happens if we look at sample size calculations? Well, this is pitiful, even in journals of high impact. Blinding is about average across the piece, and randomization, there is a significant inverse relationship between impact factor and the reporting of randomization. So when my boss comes to me and says, Malcolm, have you done anything that's any good? I'm able to say to him, great news, Jonathan. All of my work is published in journals of very low impact, at least most of it used to be, and so you can be assured that it's of the highest quality. And interestingly, if you look at this relationship over time, at the very early stages, when we've got data going back there, I think the earliest publication is 1942, the relationship is the way round that you would hope it would be. And over time, it's changed from this to this. As a community, we've become more enamored with newsworthiness, with getting into high-impact journals, with publishing and be damned. There is some good news in that this is a tractable problem. So here in 2009, we published with Uli Dernagel, who's the editor of JCBFM, Jeff Donnan, the editor of the International Journal of Stroke, and Mark Fisher, the editor of the journal Stroke, a series of good laboratory practice guidelines for the reporting of in vivo research for stroke. And this is they're published in 2009 and then adopted by those 3 journals in 2010. And this was published last year by Jens Minnerup, where he's looked at the reporting of things like randomization, allocation, concealment, and blinded assessment of outcome in the years 2010 through to 2013. And what you can see is that what you can see is an association between the implementation of that change in journal policy and an improvement in the quality of reporting. Now, whether or not they're causally linked, I don't know, but I think they probably are, and this has probably helped. And if you have journals which don't currently have such policies, I would suggest that you start to get them pretty quickly. Let's talk a bit about research regulation and delivery, because some of my work is in clinical trials. I'm one of the chief investigators of a Europe-wide clinical trial of hypothermia for acute ischemic stroke, which is regulated variously in different member states under the same clinical trial regulations as either a device trial for the cooling mechanism, a drug trial because we have to give pethidine to prevent cooling, to prevent shivering, as both a drug trial and a device trial, and as neither. And it's taken us about 3-4 years to get regulatory approval in all the countries in which we would like to recruit. So here's the delays and inconsistencies in ethics and governance with a delay. So this is 30 days here, 60 days, 90 days, 120 days for ethical approvals in different countries. You see it's getting a median of about 2 months before you can do anything. And then governance approvals, which comes on top of that, starting off at about another 2 months, so 4 months from deciding to go before you can actually go. Although in the UK now, we have systems in place with guaranteed clock starts, clock stops, which means that you should get both of those within 30 days of your application. And in the UK, about 5 years ago now, we started to set up the clinical research networks, and this is what's happened to recruitment, both to observational studies here, which are quite easy to recruit to, I think, but to randomized controlled trials has almost doubled in the UK National Health Service between 2009 and 2012. And I think that's, those of you who do clinical trials will know that the biggest challenge is actually getting patients recruited into your trials, and this helps, and it helps substantially. And so efforts like that are very much to be welcomed. Accessible full research reports, so studies published in full, including the publication of studies with disappointing or negative results. When I say this to audiences or when I say this to investigators, they say, the usual response is to say, well, we know that journals don't like negative results, and so it's their fault because we submit them and they reject them, and then we go down the impact factor scale until we get it in somewhere. That doesn't appear to be the case, actually. So this is quite complicated. It's from the Lancet series, but it's looking at the percentage of studies which are reported according to whether they're positive or neutral from the time of inception. So, sorry, from where you start to collect the studies. So if you start to collect the studies from the very beginning, 73% of positive studies are published, 50% of negative studies are published somewhere. If you look from regulatory submission, a bit later than inception, it's even worse. If you look at conference presentations, so you might see posters or platform presentations here, how many of them actually get published? Again, there's a difference with positive studies being more likely to be published. But if you look at 4 cohorts of manuscripts submitted to these high-end journals, New England Journal, BMJ, Lancet, and the like, then there's no difference between the publication rate. So what's happening here is that the investigators must be deciding not to publish. I've done my study, I've taken it to the conference, people know about it, and I can either go through the next 6 months or 8 months trying to get it published in a journal, or I can kind of set it to one side because it doesn't tell us anything, and I can move on to the next project, which will then get me a much nicer publication, I hope, which will then help me move forward in my career. So the responsibility for publication bias rests at least in part in this room. There's another problem with reporting of studies, and that's how there's a difference between what we report in terms of the benefits of an intervention or the harms of an intervention. So these are studies of one of the antidepressants, reboxetine, and comparing, if you look at the FDA portfolio for those drugs, you've got some published trials and some unpublished trials. So you can look at efficacy in the unpublished trials, the published trials, side effects, adverse effects in the published trials, unpublished trials. And what you see is that in the reported studies, there's lots of efficacy, and in the unreported studies, there's no significant efficacy. And we kind of know that that's what we would expect to see. But if we look at dropouts because of adverse effects, in the reported studies, there's no dropouts. But in the unreported studies, there's a substantial and significant number of dropouts, worse in the treatment group, favoring the placebo group that we don't know about. So there's a discord, a discrepancy between the reporting of benefit and harms. There's also a problem with the completeness of reporting. So if you go to a paper in PNAS or Nature or Science or Cell, usually there'll be 5 or 6 panels. Those panels will be different experimental tests of the same underlying hypothesis. Being very generous, each of those tests will be powered at about 50%, which means the chances of any one of them being positive is 50%. The chances of 2 of them being positive, if you've got 2, is 25%. The chances of all 6 being positive is less than 2%, but yet how many science papers have got all 6 of those falling in with a p of less than 0.05? A lot more than 2% of them. So there's a problem with what we report. And what's probably happening is that people are measuring lots and lots and lots of outcomes and then selecting for publication the ones that are most in keeping with their favorite hypothesis. So here's some evidence to support that. This is work we did with the Ioannidis group. We'd got data from about 4,000 experimental outcomes in animal models of stroke, Alzheimer's disease, and multiple sclerosis. And we knew what the overall estimate of the effect was, and we knew what the dispersion was around about it. So we could predict, with that overall estimate of effect, that of those 4,000 individual experiments, about 900 would have had statistically significant findings. And in fact, we didn't observe that. We observed 1,700 statistically significant findings. That might seem low, but remember, if you've got a dose-response curve, you would expect 1 or 2 points at the bottom to be neutral or negative anyway. So the reason then that we think we get these 1,700 isn't because the 1,700 is wrong, but because this 4,000 here is wrong. And this number should be about 8,000 experimental outcomes actually measured, of which only about 4,000 were reported. And you can calculate the percentage of suppression of experimental results, overall about 50%. But in some fields, like Alzheimer's disease research, we estimate that about 80% of experimental outcomes in animal experiments are not published, which makes it very difficult to draw any conclusions about the results, particularly in the Alzheimer's research. The most commonly used outcome is the Morris-Water maze. When we looked for Morris-Water maze outcomes, we found 56 publications. In those 56 publications, there were 59 different ways of presenting the results of the water maze, almost as if the investigators were choosing the outcomes to report because of how good it looked, not because of what they'd done. So in animal research as well, I showed you the data for the antidepressant earlier on. In animal research as well, you get different patterns of publication and outcome reporting bias in different fields. So this is looking overall at our experiments that have been testing the effectiveness of a drug at improving an outcome in an animal model largely of neurological disease. And you can see a reported improvement in outcome of 40%, but when you correct for publication bias, that reduces to about 30%. There's another experimental paradigm in animals which is often used, and that's the reproductive toxicology paradigm, where you take a pregnant dam and you expose her to a chemical that you would like to use in your manufacturing process for the plastic milk bottles that we make up baby milk in to give to little Johnny. And of course, the Dow Chemical Company need to show that's safe before they can unleash it on an unsuspecting population. So what you do is you take a pregnant mother, you expose her to this chemical, and then you measure the change in birth weight of the pups that result from that pregnancy. Now, notwithstanding lots of problems in that literature around pseudoreplication, there's another problem, which is that if you look at the reported change in birth weight, the reported change in birth weight is a reduction in birth weight of about 0.3 standard deviations, standardized units. So that's a fairly small difference, but when you correct for the presence of publication bias, the difference is actually substantially and significantly higher. So the publication bias goes in the direction of the interests of the people who are doing the experiment. If I'm a drug company or an investigator and I've got the next new drug for stroke, my publication bias will favor the drug. If I'm trying to prove that something isn't happening, adverse effects in animals or in humans, the publication bias will go in the other direction. Now, I talk a bit about the problems of sample sizes and small sample sizes, and people aren't altogether clear as to why this should be a risk of bias. When we look at our data in animal models of multiple sclerosis, we find an interesting relationship between the size of the groups, 1 to 4 animals per group here, more than 10 animals per group here, and the estimates of how good those drugs are. It's a strange phenomenon whereby the smaller the study, the larger the effect. And you could say, well, that's a consequence of the power calculations that were done, but we know that power calculations were certainly very rarely done, never reported, and doing an experiment with 1 to 4 animals per group is kind of crazy anyway, unless you're really, really silly. So we tried to work out what might be underlying this. So we did some simulations. So we said, let's assume that we've got a true effect size of 10 units of whatever, with a standard deviation of 10, and let's sample some experiments with different group sizes and see what happens. And let's assume that only those experiments which reach significance in a t-test at p of less than 0.05 get through to publication. So here's what we get. With 5 animals per group, only 30% of studies get published. With 20 animals per group, 86% of studies get published. This is a bootstrapped power calculation for the cognoscenti. But what's important is that if we take those 30%, we say, let's take those 30% of studies that were published and let's meta-analyze them to see what they say. And this is what happens. The effect size in the small studies is substantially and significantly higher, and it falls away as group size increases for the same underlying biological effect in the presence of publication bias. And the thing about, which to me is quite frightening about this, is even with an adequately powered study, there's still an inflation of effect size in place where there's publication bias. So how does that work? Well, let me try and explain it. So you've got 2 cohorts of studies here. They both have the same central estimates of effect. These ones are underpowered. These ones have got better statistical power. So if we assume that this one here isn't, if we then summarize them, we get the same overall estimate of effect. But if this study here isn't published and we don't know that it exists, then this is what happens, and we see a difference between these that isn't really there. So although small sample sizes per se don't have an effect on your experiment, apart from wasting your time because you're underpowered, they do pollute the literature because of the mediating effects of publication bias. Let's look now at unbiased and usable reports of research, and for me, certainly in animal research, this is becoming absolutely critical, and it's critical to what's become known as the replication crisis or the reproducibility crisis. Steve Goodman and colleagues recently came out with a schema for thinking about what replication and reproducibility is, which I think is quite helpful. They say that they've solved the problems, and these are very discrete, but we'll talk about that in a minute. Methods reproducibility is the ability to implement as exactly as possible the experimental and computational procedures with the same data and tools to achieve the same results. Results reproducibility, the production of corroborating results in a new study having followed the same experimental methods. Inferential reproducibility is making of knowledge claims of similar strength from that replication or reanalysis. You can imagine that 2 investigators with a different underlying hypothesis might look at the same data sets and come to different conclusions. Robustness, the stability of experimental conclusions to variations in either baseline assumptions or experimental procedures, and generalizability, the persistence of an effect in settings different from and outside of an experimental framework. Very flatteringly, I see someone with a camera taking photographs of my slide. Those of you on Twitter, if you look at the ECTH 2016 hashtag or thingy, whatever it is, will find my slides in Dropbox there, and you should feel free to use and abuse and adapt and adjust them as you like. So looking at all of this, they say it makes things a lot clearer, but I don't think it makes things completely clear now, because we've still got a continuum between methods reproducibility, trying to do exactly the same thing, results reproducibility using the same methods, and I'm not sure of this distinction, and robustness when we vary these things slightly. So we should be moving along this pipeline, along this passageway. So these are data from 1999, when Jim Crabb was interested in whether he could do the same experiment in 3 different labs and get the same answer. So they used 8 different strains of animals which originated from the same animal house. And they were sent out to Portland and to Edmonton and to Albany, and they did exactly the same 8 behavioral outcome measures using the same protocols, the same tests, everything. And 2 of the strains of animals they used, these are a 129 background, and this is a 5-HT1B transporter knockout animal. And the outcome here doesn't really matter, but it's the number of total entries into the open arm of an elevated plus maze. And just to simplify that diagram for you, what they showed was that in Albany, the transgenic was significantly worse. In Portland, it was significantly better, and in Edmonton, actually, there was no difference between them. So you're getting completely contradictory results trying to do exactly the same thing at the same time from the same protocol. And in fact, when they looked into the detail of the amount of variance that they observed that was explained, they found that there was an effect of the genotype. There was a site effect for all bar 2, which explains sometimes as much as 30% of the observed variance. There was an effect of sex, an effect of whether the animals were done straight off the plane or whether they were bred for a few generations before doing. Interactions between the genotype and the site, between the genotype and the sex, no interaction between genotype and shipment. So you're explaining a huge amount of the difference between findings in different places on the basis of something other than the underlying biology which you're studying. And Bernard Vokel and Hanno Verbel have published this in Tips just a couple of months ago, and this is well worth reading. They talk about reaction norms, and the idea is that you've got a response here that you're interested in, and you've got some underlying biological thing, which is a nuisance variable, which is impacting on your results. And some people might be doing an experiment down here and getting this result, and some people doing an experiment down here and getting this result, so these results are contradictory. But they're actually measuring the same underlying biological phenomena. It's just that they're at a different place on this reaction norm response. And of course, as a clinical trialist, I'm very interested in this because I want to do a clinical trial of a drug that works all the way along this if I can. I want something that's got widespread applicability. So you can imagine that there's more than one nuisance variable. There are many more than one. And you can have a kind of map of where the efficacy is and where it's not. And so looking at animal data to take into clinical trial, firstly, I like to see that there are limits to efficacy, because then I believe the data. But I also like to see a situation where the top of this surface plot is a very gentle hill, rather than a very tightly sloping situation where you only get efficacy under very particular sets of circumstances. So generalizability is very important. And to understand generalizability, you've got to understand how the experiment was done. Goes without saying. Here's our MS literature in animals again. We identified over 9,000 publications. We were particularly interested in about 1,000 of them, and even more interested in 203 publications that were describing the effectiveness of 36 particular drugs of interest. So we went to those 202 publications. We had to exclude data from 72 of them, for very simple mathematical, it wasn't that they didn't tell us stuff about the experiment, it's that they didn't tell us the number of animals per group, or the variance, or both. Three of these 203, we didn't know what the paper said. I took these papers to 2 professors of neuroimmunology who were expert in the animal modeling of MS and say, what does this say? And they came back 2 days later to say, I simply don't know. Nothing useful. And so for 1/3 of the publications involving animals there, nothing useful to be found. Now, I've talked a little bit previously about the Morris water maze. Does anyone know the Morris water maze? No, you're all Morris water maze virgins. Okay, so you want to know how good an animal's memory is. So you've got a pool of water, and you make the water milky so the animal can't see anything. And you've got a load of social cues around about the room. So depending on who the lab technician is, it might be One Direction or Elvis Presley or Angela Merkel as what they've decorated their room with. And the idea is that there's a platform which sits just underneath the surface with a flag to say where it is. And in the training days, you put the animal into the opposite, I was going to say the opposite corner, the opposite quadrant of the water maze. And the animal eventually learns that if it swims to the flag, it gets out of the water, and it doesn't like being in the water. So that's what it does. And how well the animal remembers where that is in relation to the cues is tested the day that you take the flag out and the animal swims to where it thinks it remembers the flag was. And if it gets there, there is a platform and it gets out. So there's training days and test days. Firstly, this is the size of the Morris water maze pool. These are experiments done in mice because there's transgenic in the old days. And you can see the pool size ranges from 85 centimeters to 2 meters. This is putting a mouse into a rat pool, but in fully 20% of the studies, it doesn't tell you how big the pool is. So that makes it difficult to replicate. This is the temperature of the water, which contributes, of course, quite substantially to the versive input to the animal to get it out of the water. So this is Scotland on a very good day in summer, 16 degrees. And this is where I would like to be able to go on holiday if I had enough air miles. And you can see there's a huge range, but in 40%, it doesn't even tell you how warm the water is. And I told you there's training days, so you've got a number of trials per day and a number of days training. And the modal response is it doesn't tell you either the number of acquisition trials per day or the number of days training. So it's impossible to replicate this work because you don't know what they did in the first place. I would put these problems with research not under a heading of fraud, because I don't think that people are deliberately making up their numbers. Research fraud is rare. It's not quite as rare as I think we would all like to believe. I've had now to adopt a practice when I see papers for review of copying the Western blots into Adobe Photoshop and turning the contrast up and down to see which lanes have been cut out of another gel and stuck on. And there's lots of entertaining things on the internet to look at about that, and some PubMed Commons comments on that, and some histochemistry as well. But there are also dubious research practices where we do things not quite right, and we know they're not quite right, but we do them because they seem to work for us. So we use experimental designs where there are opportunities for the investigator to influence subject selection. We'd get around that by having proper randomization. Where there are opportunities for the investigator to influence the conduct of the experiment, which we would get round by blinding the conduct or the assessment of outcome. Often we don't have a stated group size, and often we're quite flexible in how we add to the groups as we go along. We might get to 0.08 in our first analysis with 8 animals per group, and our boss might say, just stick in another couple of animals and rerun the analysis. It's significant, it's just not significant yet. P-hacking, which is the practice of throwing any number of different statistical tests at a problem until you get the result that you want, and harking, which is the delightful process of hypothesizing after results are known. So I reviewed a paper 6 months ago, and I said, well, nice data, but it doesn't confirm the hypothesis that you say it does. And a week later, the reply came back, we're very, very grateful to your esteemed reviewers for their thoughtful and insightful comments, that's what they always say, and we completely accept what they say about our hypothesis, but we've changed our hypothesis, so now everything's fine. That's not quite the way that hypothesis testing research should work. So I think we need, as a community, to be better at distinguishing between exploratory and hypothesis testing experiments. For exploratory experiments, don't use hypothesis testing statistics. It seems kind of self-evident. It's fine to be exploratory. We need exploratory research, because if we didn't have exploratory research, we wouldn't have hypotheses to test. But don't do statistical tests on exploratory data. For hypothesis testing experiments, specify what you're going to do in advance. Register your study so that people can see what you asserted you were going to do in advance and that you actually did it. And do a sensible sample size calculation guided by a positive predictive value, not a p-threshold. Let me briefly tell you about that. So what can go wrong with an experiment is that the effect might be there, but you don't detect it, a false negative, that your test falsely calls an effect, a false positive. Now, with our conventional p-value of 0.05, what that actually means is the probability of getting that result or a more extreme result in the situation that our hypothesis is not correct. It's not the chances that our finding is incorrect. So let's assume that 20% of hypotheses in a field are correct, should be about right. We're powered to detect a biologically important effect of 20%. That comes from the neuroscience literature, and that's been demonstrated. And let's take a critical p-threshold of 5%. So for every 100% of studies, 20% will be truly positive, 80% will be neutral or truly negative. If we are powered at 20%, 20% of 20% or 4% will test positive, 16% will test negative. If we've got a p-value of 0.05, of these 80%, 5%, which is 4%, will test falsely positive, 76% will test negative. And that means that faced with a positive test result, actually, 8% of our results are positive, and they're equally distributed between the truly positive and the falsely positive. So the positive predictive value of this experiment, powered at 20% and with a p-value of 0.05, is no better than tossing a coin. And so what we should do is think about having, in the same situation here, 20% of hypotheses in this field that we've made up are correct. We're powered to detect a biologically important effect at 80%, not at 20%, and let's take a p-threshold of 0.01, and then what we see is that 16% of studies are truly positive. We still miss 4% of the positive effects. We get far fewer false positives and a positive predictive value of 95%. And if you're going to do a piece of research which is going to change the way that we think about the world and we think about biology and we think about science, I think this is the standard of proof that we should be aiming for when we design those experiments, rather than sitting in our laboratories and tossing a coin. I said that we should say in advance that what we were going to do and make that available somewhere that someone can see it. It can be closed behind some blind link on the Open Science Framework or somewhere, and we just say to our peer reviewers, here, if you go here, you'll see that we deposited this protocol 2 years ago. Now, as a clinical trialist, I've often got nothing to do except write my clinical trial protocol. I've got 10 years to do it and get it published. As an animal researcher, where the cycle of an experiment is months, not decades, that's different. So we're advocating the idea of very brief 6-line protocols, rather like a haiku, population, hypothesis, intervention, sample size calculation, primary outcome measure, and statistical analysis plan with a line for each. And my rationale for that is that if you show me that you said those things 2 years ago or 6 months ago before you did your experiment, and that's what you actually did, my confidence in your results goes way, way, way higher than it is if you can't demonstrate that you did what you set out to do. You should allow others to check your work, making all your data available, even if you need a sunrise clause. So these are our data from our PLOS biology paper last year. They're all there for people to check. These are data on Dryad from, I think, our Alzheimer's study. And in fact, people have done independent replications of some of our meta-analyses, and it's not very worrying, but it's quite perturbing when that happens. But it's fantastic when they come to the same general conclusions as you did, and it's very good to be able to do that. I think there's a thing that we're not doing that we should do, and that's we should do research improvement activities. Those of you who are clinicians will know about healthcare improvement strategies in the Healthcare Improvement Institute. We should do the same thing with research. And there's a number of different ways that we could do this. Nature changed their publication guidance 2013 for in vivo research, and we're doing a study with Nature, a before and after study, matched with similar publications in other journals, to see whether their implementation of this policy has changed the quality of what they report. With a major publication which cannot yet be named, we're doing a randomized trial of whether forcing people to complete an ARRIVE guideline checklist improves the quality of the research. And we're now in the stage of outcome assessment for all bar the 10 papers that still have to have an editorial decision. So we randomized about 1,600 publications, and we're looking at the quality of reporting in the 700 or 800 that have been accepted for publication. And we'll see if it makes a difference. And here, for institutions, I think that an institution, for instance, where only 7% of the work was reported to be randomized, might like to think about having an institutional audit and improvement activity, planning what to do, actually doing it, studying what the results were, and acting to see whether you need to embed that into current practice. So I wouldn't start from here if I was trying to do it, but you do a PDSA cycle. If you find that something does improve things, you implement it into your standard institutional frameworks. And then you try something else, and if it works, you implement that, and then something else and implement that. So gradually, you get to improve things. The National Centre for the 3 Rs in the UK has got an online experimental design assistant, which is slightly clunky the first time you use it. It's very easy to use the second time you use it. Will provide a critique of your experimental design and your analysis plan, and will give you a dated PDF of what you said you were going to do at the time that you could then send as with your letter to the editor. We've got screening platforms for systematic reviews, shortly to involve a little machine learning bit behind this to make the review happen more smoothly. And we've got training in the critical appraisal of publications in biomedicine through this Nature study, because if you participate in the Nature study, first of all, you've got to be trained, and there's training materials there, and then you get a certificate saying that you know how to critically appraise a paper, which may be a useful thing to give to your PhD supervisor or your boss the next time you get appraised. And if you actually get so far as participating in scoring some of these for outcome, anyone who contributes even one measure to the outcome gets their name as a collaborator on the paper. If you do 10, you get a natty little USB stick with our name on it, so I'd highly recommend it. None of what I've told you today would really have been possible without the approach of systematic review and meta-analysis of animal data. It's involved over the last 10 years collecting large amounts of data, not just now in neuroscience, but beyond into cancer biology, myocardial infarction, cardiology, and other areas. And if in your areas you're at all interested in doing a systematic review of animal studies, please let me know. And we've got people all around the world who do it without having to come to Edinburgh, although you might like to come to Edinburgh. And you can do it from a distance, and I think it will help inform you about your work and how you can make it even better. Thank you again for the invitation. Thank you very much for listening.
Professor, on behalf of, I think, everybody in the audience, thank you very much for this wonderful talk. Not only giving us insights into the problems, some help perhaps, but also knowledge now of cool terms like harking and FLUSP, I think is now the new term to plan our studies. But on behalf of the ECTH, I would like to thank you once more. And with that, up to the next session. Thank you.
Thank you.